So it means that we can use differentially expressed proteins for enrichment analysis to get further insights that which type of proteins are involved in which type of functions. Welcome to another video tutorial. In this video, I will tell you how we can interpret the results of proteomics data. So before we talk about the further detail of proteomics data, let's talk about what is proteomics. As we already know about the central dogma of life, that first DNA is changed into RNA and then RNA is changed into protein. So if we study the whole genome, that would be called as genomics. If we study the whole of transcriptome, it will be called as transcriptomics. And if we study the whole of proteins, any individual, it would be called as proteomics. These days, proteomics is being commonly used to address many biological questions. Let's take one example that you have two tissues, one of them is normal and other is diseased, and you want to know that which type of the differentially expressed proteins are causing that disease in that tissue. So how you will address that question? First, you will take those tissues, and from those tissues you will extract all of the proteins. And as we know that all of the proteins cannot be directly fed to the LCMS because of their large size, so it is important to first break down into their peptides, and then peptides can be easily fed to these LCMS based systems. After that, we deploy different genomes and find that these peptide sequence match to which type of the peptides, and then we found their assembly, and after that we done their function analysis. That is why we call it as bioinformatic analysis of the proteomics data. So how we achieve this through our workflow? So first we should have a source of protein, then we should extract those proteins and we should digest them to get their peptides, and then we should separate them in the form of LC. We know that LC is a liquid chromatography and it is a separation technique, and we combine this LC with MS. So as we have now two separation media, so that is why we also called it as the tandem mass spectrometer. So if we open this mass spectrometer, what happening there? Here there is one ionization source in which the samples are being propelled through this mass spectrometer, and you can see here these peptides and these amino acids have different ions, so which are being deflected with the help of these different magnetic charge and magnetic fields. After that this magnetic acquisition is transformed into the form of spectra. These curves are also called as spectra. So after that we only extract the useful spectrum and we discard the remaining. So this was the simple protocol and simple workflow which we apply through proteomic study. Here are the commonly used proteomic services nowadays. Although there are the many types of proteomic services are being commonly used in different studies, but among them label-free quantification method is being mostly widely used because in this we don't deploy any radioactive material or radioactive chemical, so that is why this is cheap and less time consuming. In almost all other methods like ITRAC, TMT, SILAC, and absolute quantification method, semi-quantitative proteomics analysis, we apply different radioactive chemicals. So that is why, because radioactive chemicals are expensive and difficult to handle, so that is why all of these methods are radioactive based, and only this one method is non-radioactive based. So I will not cover them in detail because the focus of this video is only about how we can interpret the results of proteomics data. This thing I will cover in separate video. So here is the first visualization of proteomics data which we achieved through. As we know that usually whenever we fed the peptides through this mass spectrometer, we get the data in the form of chromatogram. So if we look carefully at the chromatogram, we can see usually a chromatogram has two axes. You can see on the x-axis is retention time, and on the y-axis its intensity. Here is one curve in blue and another one is red. In we can see here in the blue, this is the total ionic chromatogram, and in the red this is the base peak intensity. And here you can see in the whenever we plot the chromatogram, the first few hours and the last few hours are empty, because here we all of the mass spectrometer need their loading time, so we usually discard this for initial and the last part in the proteomic study. As you can see, this chromatogram is so messy, so it is almost difficult to extract the different proteomics or peptide based information. So it is important to extract only the relevant information and discard the remaining. So how we do? We achieve this through the extracted ionic chromatograms. Now we have discarded some of the length and we have only retained only important length. Later on I will discuss how we do this. So other things are same. Here you can see here is intensity and here is retention time, or we have only keeping few curves and neglecting other curves, so it would be called as extracted ionic chromatogram. In real life, whenever we achieve this, data is in look like in the form of these 2D graph. Here you can see this is the ms1 map. In this, retention time is plotted on the y-axis, and on the x-axis you can see here the ions are separated on the basis of their mass to charge ratio. And here is the 2D map, heat map of ion mobility. Later on I will also discuss that how this ion mobility heat maps are generated. We have mass to charge ratio, and here we have inversed ion mobility. In reality, these chromatogram usually look like this, but we process these chromatogram in the form of this. Here are the common types of peptide fragment ions. To understand the common type of peptide fragment, first we have to understand the structure of any amino acids. We know that usually amino acids are linked, these form into peptides, and then peptides together make proteins. What is the shape of the amino acids? You can see here usually any amino acid has its amine group on its side. Then they have carbon which is attached to the carboxyl group. Then it is also linked with R chain. Actually, this R chain varies amino acid to amino acid. Here are the different types of amino acids. The basic structure, here is the amine group and the carboxyl group. Only the side chains are different. So it is important to know that as this R chain may varies from amino acid to amino acid, so the charge on these amino acids also vary from amino acid to amino acid. What I want to convey from this busy slide is only this, that some of the amino acids are positively charged, others are negatively charged. Similarly, some of the amino acids are polar and other type of amino acids are non-polar. So whenever we pass these amino acids through any of this mass spectrometer, we usually know that whenever Each amino acid is linked with another amino acid. Usually we have a water molecule, and then this amine group is linked with the carboxyl group of the first amino acid, and new dipeptides is formed. So whenever we passes through these amino acids long chain through the mass spectrometer, so it is important to know that whenever we deploy the different type of energy, different length of bond breakage is also possible. So it means there would be also the different types of the fragment ions. So some of them would be useful and some of them would be non-useful. So it is important to keep only the useful, because the non-useful we don't need. Let me show you how we do this. So here is the visualization of proteomics data at the fragment ion level. How we achieve this through, let's suppose this is a peptide. You can see here are the different amino acid N, T, I, N. If let's suppose if we have a protein, let's suppose my name is like Asif. If we have a protein who have these amino acids, so how we can separate this? So the first amino acid, let's suppose, is D. So how we can achieve this? The first would be D, and the second would be the D and R. Let's suppose the peak of this is here. The next one is D, R, and A. The peak is there. Now determine the difference between their mass to charge ratio. So the third one would be here. So now we can know their mass to charge ratio difference. Similarly, you can see here whenever a protein has its amino acids, so we can separate them on the basis of their mass to charge ratio. So we achieve different mass spectra, whether it is data dependent acquisition or data independent acquisition. Here we have the mass to charge ratio, and again we have the intensity. It is important to know that how we change this data dependent acquisition of spectrum to their relative intensity. As we know that it is possible to collect the intensity of peaks in the form of their ions. So here we change their spectrum according to their relative intensity, and we know that mass to charge spectrum reflects also the ion abundance. After we achieve the mass to charge spectrum, which is reflecting the ion abundance, we will get the illusion profile in the form of these peaks. And here it is important to know that we are discarding few peaks, and here we are keeping few peaks or illusion profile. So how we achieve this? Here we have in the blue color we is precursor, which is matching with the sequence of Y5. How this we achieve? We compare this illusion profile with the help of online databases. What we do actually, we change these profiles and to their amino acid sequence, and we compare them with the help of online databases protein sequences, which is achieved through the decoy. With the help of deploying these decoys to these illusion profiles, we can keep few important precursor and we can discard the few remainings. So after that, how we change these fragments to their peptides? You can see here, normally the peptides are visualized in the form of two ways. One of them is uncollapsed and the second way is collapsed. So here you can see, here let's suppose this is our database sequence of proteins, which is achieved through the decoy. This is the sequence from the database, and those illusion profiles here are matched with, like, this part is matching with this one, this part is matching with this one, and this part is matching with this one. Uncollapsed peptide coverage plot can also help us to know that which part or which fragment is being accumulated many times. So another way is through this alpha map peptide coverage plot. Here you can see, now let's suppose this is the sequence of protein in the database, and this is the sequence of that our matched alleviation profile. This is the matched peptides. Note that there is no gap between all these peptides because based on the database sequence we have just joined together to get a final protein. The mutant and the wild type coverage. Let's suppose if you have a protein sequence, and here you can see in the wild type there are these three fragments, and the mutant type we can find few more addition or deletions. In this way, the peptide mass spectrometry can also help us to visualize the peptide sequence. Mass spectrometry data can also help us to visualize the peptides which are being phosphorylated. So how we achieve this? So to understand this, let's understand a hypothetical example. Let's suppose we have a protein whose position on 0 to 1 200, and from 200 to 400 there are the different types of amino acids which are being phosphorylated. Here are the different positions of amino acids which are being phosphorylated. So we can combine this information with this online databases, and we can know that which type of the amino acids have more probability of phosphorylation. The size of these lollipop circles are showing the probability. The bigger the size, the higher the probability. So it means that we can also use these peptide visualization data to get the phosphorylation sites. Now we are approaching to the protein quantity visualization and basic analysis. Probably this type of the figure you have often seen in many manuscripts. So here up we have the different intensity peak graph. The wild type has three replicates, like this is R1, this is R2, and this is R. And similarly here is one mutant type which has similar peaks. Within these replicate, these peaks should be same. It is also important to understand that why here the peaks are so messy. To understand this, first we have to understand that, like, if we pass through the carbon element through the mass spectrometer, what happens? Actually, the carbon has different isotopes like C12, C13, and C14. So actually it can all differentiate these C12, 13, and 14. So that is why it is important to keep only required one. Let's suppose we are only dealing with C12, so we have to discard C13 and C14. Peaks which is deployed in the form of log 10 intensity is consist of isoforms and paralogs. The second important point, that whether there is a range and reproducibility in the data. So if your replicates have similar trend, so it means your data has reproducibility. If you see that there are different intensities within these replicates, so then we have to deploy the different normalization. After we have applied the additional normalization, so we can transform these peaks into the different rank plots. Let's suppose this wild type peak, how we will change them into the rank plots. It is important to note that which type of the relative abundant proteins should be typically included for our main data analysis. So if we transform these proteins in the form of this, so first we should note that here what type of proteins are normally found, and here what type of normally found. So you can see here the color shows these are actually mostly the structural component of different ribosomes because they are abundant in nature. Our main data of different abundant proteins would be available here. In the green color, it is showing the different transcription factor and different signaling proteins which are essential for any biological question. If we are comparing the two conditions, like here we are comparing the wild type and mutant type, and in this our example the mutant type is RPN4. Now this is a time to compare the correlation of with 3R with 1R. Similarly we have now with 2R with 2R, and similarly with 2R with 3R. So if you have only two samples, like here in our example we have wild type and the mutant type, we have RPN4. So if we are comparing the sample with at only two conditions, then we can draw the graph like this and we can find a correlation between the different time course. But what happen if we have more than two conditions? Then we have to draw these heat maps. Here let's suppose like now we have wild type on single side, and here we have also the mutant type on single side. So now we are comparing at least four conditions. So it means because here we have two conditions of wild type and we have two conditions of mutant type, then we need to draw the heat map. And if we try to look further, you can see here within this heat map this condition is similar to like this one. Here the same color is showing that they are positively correlated, and here within this blue color they are positively correlated, but among them they are negatively correlated. And here you can see this correlation curve is showing where the color will be yellow, it is showing the positive color. Similarly like this one, here you can see here is the positive one correlation, so here similarly here is the positive correlation. Within these boxes color should be the same, and out of these boxes it should be the different if there is a differential expression of proteins. While moving to the next, protein quantity visualization and basic analysis, you have often seen these type of the volcano lines. So how we can understand if we are comparing the two samples, we can present the data in the form of these values. And as we know that we will be unable to get any differential proteins until we have applied any statistics. So here we have two types of these statistics. So the first one is the power statistics and the second one is the S0 parameters. So what we do, we usually apply different fold change. Here you can see this and these are the significant, and here in the blue color these are the non-significant. So if you want to find the significant proteins, you can positively and negatively, because this here shows the FC value is in negative and here the FC value is in positive. So these are the positive significantly differentially protein and these are the negative significantly differentiated proteins. Can also apply this S0 parameter, which is also a form of another. Similar to these power statistics, we can also apply this S statistical analysis. Here we have positively significantly correlated and here we have negatively significantly correlated protein, and here we know that each dot is representing one protein. As we know that here each point is showing one protein, so we use different enrichment analysis to find that these type of the proteins belongs to which pathway. We have the negative log 10 p values, and similarly these proteins are related to which biological function. The yellow color is showing the odd ratio, and similarly the size is showing the significance. The bigger the size, the bigger is the number of the significant proteins. So the highest number of the significant proteins were related to the protein refolding. The proteins which are related to the proteasome mediated ubiquitin dependent protein catabolic processes were also significant. So it means that we can use differentially expressed proteins for enrichment analysis to get further insights that which type of proteins are involved in which type of functions. Previously, we have dealt the simple example where the number of treatments was only 2. But what about when the number of the treatments are more than 2 and involve complex design? So you have often seen these type of the PCA analysis in different complex experimental design. So it is important to note that why we use the PCA analysis, because we use the PCA analysis for dimensionality reduction. How we can understand this? Let's suppose this is a plane and this is treatment 1 sample, this is treatment 2, and let's suppose these samples are very different than T1 and 2 and lying in another plane. So it means if we don't use this dimensionality reduction, it is almost impossible to draw all of these samples within one plane. So we use this PC analysis, so which can mask the correlated and orthogonal effects. And here the each color is in fact showing one treatment. So whenever we have the PCA1 and PC2, all of the samples are grouped together and clustered together. Here you can see the legend is showing at the different 0, 3K, 6K, 12K, these are the different names of the sample, and you can see within these treatment they are clustered together. But it is also important to note that PC1 always have high variability. You can see PC1 has 63% and PC2 has only 20%, and similarly the PC3 has more or less, and similarly PC4 would have even more less. So it means PC1 is always higher than PC2. You may also notice that this type of the principal component analysis we often observe in the transcriptomic data. But what about if there are more outliers? Then we applied two different algorithms. One of them is TSNE1 and UMAP1. How they are different than this PC1, you can see here these these are more better reduction algorithm when there is more outlier. With the help of this TSN1, we can find more nonlinear obvious separation. Here the many of them were clustered together, but when we apply TSN1 we have more separation. You can see here we can see more separation than this. Similarly, UMAP reduction algorithm can help us to also know better about the time course experiment in the different organelles. Now we will move to the common network types we encountered in usually proteomic studies. So here is the first one is in the form of the heat map. So you can see this heat map is also showing with its marginal dendrogram, which is actually showing the sample similarity distance between the protein quantification. In this, each color is representing one treatment or one sample, and here each sample has been shown with its different color. Color is representing the normalized z-score profile. So if the color is blue, so it means the expression is positive. You can see this sample and this sample is showing blue color, it means this is showing the positively correlated. However, this sample and this sample is showing the red color, it means the expression of these two set of protein is negatively regulating. So it means in this is showing negatively correlating. So you can also visualize your individual dimensions of data and samples through this heat map. Another way to represent your proteomics data is through these line plots. You can see here the line plots also shows the normalized profile at different time scores like 3K, 6K, 12K, 24K and 80K. Sometimes we often come through a situation when the dimensions of different samples use the different time scales, like when we are dealing with different parameters, like one parameter we are measuring like is height, the second parameter we are measuring is related to the quality. So it means that these both parameters are not related to each other. So then we need to find a statistics that is called Q value, and we know that usually Q value is used for finding someone is whether a good fit or bad fit. So another way to represent the normalized expression profile when you are dealing with more than two treatments is through this radar plot. You can see here we have different time like 3K, 6K, 12K, 24K and 80K, and here each color is showing the different compartments like endoplasmic reticulum, endosomes, Golgi bodies and different lysosome. So whenever we change the time, the location or the compartment also change. So when we are dealing with the different time course and the organ tissue, we can represent our data through these radar plots as well. So now we will move to the next part, which is the common network types encountered in the proteomic study. So we know that we can usually form these interaction networks through different software. So one of them is the string, which form the network on the basis of their literature, and sometimes we also make these interaction using this reactome, and we can also use this cytoscope for different interactomics. In order to interpret the information from these network is really tricky. Like here you can see in this weighted network interaction, you can see this protein, like this protein, each protein is connected with many other nodes. There is no exact and here you can see that each node is connected with many other different proteins, so it is almost impossible to get that which protein will act as a central part of interaction. But if we talk about the hierarchical network interaction type, here we have one central protein or central node which is connected with to its daughter nodes, and then these nodes are connected with further protein. These type of hierarchical network interactions are helpful to get any centralized protein to know that which type of the other proteins are interacting with your candidate protein. The third type of interaction is the semantic pathway. Here some proteins are blocking the expression of any other protein, and these proteins are indirectly also affecting the protein expression and protein interaction of other nodes. However, all of these interaction networks are only semantic and cannot be used until further validation through experiments. With these common network types I have discussed in the proteomic study, I would like to stop here. If you have still any question related to the proteomic studies and its interpretation, I will paste the link of that review article in this video description. And if you have still any question related to this video, please let me know in the comment section. Thank you for watching. Bye bye.